In this video, we will be going over the common math equation and conversions that are needed for this molecular and cellular biology lab. We will start off with metric system. This is something that you would have already been exposed to in the GB1, GB2, and GB3, so it should not be new to you. It is an express unit of measure that is based on a base 10 decimal system. This allows us to convert from smaller to larger values simply by the movement of the decimal point. Each measurement, it has a base unit, and then you have prefixes that we can add to the base unit in multiples of 10 as needed. The biggest advantage of using metric system is that you can convert units simply by moving a decimal place without any complex calculations. It is universal and it is used by all scientists around the globe to communicate. The most common measured quantities are distance, volume, mass, and temperature. Each one of them has a specific metric system base unit attached to it. For distance, it is meter. For volume, it is in liters. For mass, it is in grams. And for temperature, it is in Celsius. Over to the left side, you can see prefixes that we would use in case we wanted to convert from that base unit into a larger or a smaller value. To convert between the numbers, you simply you can use this mnemonic: King Henry died by drinking chocolate milk, to convert from the base unit up or down the decimal system. So if you wanted to convert grams into kilograms, you would simply, if you had 1 gram and you wanted to convert it into kilograms, you would simply move the decimal point to the left 3 times, and you would end up with 0.001 grams. Since it is 1,000 grams would equal 1 kilogram. So in metric system, if we were to do that, we would simply add that prefix to the back, and it would be kiloliter or kilometer or kilogram, whatever it is that we are converting it into. So here are some practice problems for you to do. The first one is 1.5 ml is equal to how many microliters? If you said 1500, that is correct. 100 microliter is equal to how many ml? And it should be 0.1. 0.06 ml is equal to how many microliter? It should be 60. 250 microliter is equal to how many ml? 0.25. And 0.003 ml is equal to how many microliter? It should be 3. Next, we're going to go over making dilutions. When we make dilutions, we make weaker solutions from a stronger stock solution of some sort. For example, you can have a frozen concentrate of orange juice, and you can dilute it to make your final orange juice that you can drink. It is expressed as a volume of solution being diluted and the total final volume of your dilution that you prepare. For this one, you do not have any units. You would just use cans or milliliters or parts, and then you would just be expressing them as one number to another, like 1 to 10 or 1/2 or 1/4, whatever the case may be. If you use 3 parts of water and 1 part orange juice, for example, then the dilution would be expressed as 1 out of 4 or 25%, since you had 4 total parts and 1 of those was made of orange juice. When we do that, we need to understand what the dilution factor of that dilution is, because many times we use the diluted samples to run our experiments, but then before we have our final answer, we need to convert that back into the unconcentrated form. So we need to know what the correct sample would be. In this case, our dilution factor would have been 4, since we had 1 out of the 4 parts was orange juice. This is used in our labs, like I said, to correct for having a diluted sample during the experimentation. So once we get the answer, we multiply by the reciprocal of the dilution that you had made, which was the 1/4. So you would multiply the answer by 4 to get the correct answer. To prepare volume-to-volume dilution, we take the appropriate parts of each to mix them together. So in this example, we have a 1 molar stock solution, and I want to prepare a 1 to 10 dilution of that stock. To do that, I would take 1 ml or 1 parts of the stock solution, and I would add 9 parts or 9 ml of water to get 0.1 molar final concentration of my stock solution. Here are a couple of problems for you to try. Let's say that you have 100 ml of a 5 molar solution and you want 100 ml of a 30 millimolar solution. In that case, how would you do that and what would you get? Any time that we need to use to make a calculation like this, we use an equation C1V1 equals C2V2, where the beginning C1 is the starting stock concentration of your solution. V1 is going to be the starting volume that you added off that stock solution to prepare your dilution. And on the other side, you are going to have your final concentration of your working solution and the final volume of the working solution that you prepared. So in this case, if we do that, 5 molar is the starting concentration of our stock solution, so that would go for C1. We do not know the volume of the stock solution that we used, but we do know the ending concentration of our solution, which is 30 millimolar. Now this number is different than 5 molar. So you need to either convert 5 molar into millimoles, or you can convert the 30 millimoles into moles so that the units are the same. In my case, I chose to convert from millimoles into moles. So I got 0.03. And I multiplied this number with the final volume, which is the total volume of dilution that I prepared. So I prepared 100 ml of the 30 millimolar solution. So you would express your equation just like this, and then you would solve it out. When I solve it out, I get 0.6 ml of X. X, as you know, is the volume 1, which is the amount of volume that you take of your stock solution to prepare your final dilution. 0.6 ml would be the amount of stock solution that you would take. Since you need to prepare 100 ml total volume, you would then add 99.4 ml of the diluent to your mixture. Here's another example. In this case, I have an aliquot of 5 ml of 5 molar solution, and I've added that to 995 ml of a diluent, and I want to know my final concentration. So again, I would work out which one of these numbers belong where in my equation. My stock solution is 5 molar, so that goes for my C1. I know how much of this stock solution I added, which was 5 ml, so that goes for V1. On the other side, I need to know this concentration is what I'm trying to calculate. I need to know the final volume of my dilution. I know that I added 995 ml of the diluent and I used 5 ml of the aliquot of my stock. So the total volume of my final dilution is 1000 ml. I solve it out to get 25 molar over 1000 or 0.025 molar, which is a small number, so you would want to convert it into a millimole instead, and I get 25 millimoles of my diluted mixture. That is the concentration of my diluted mixture. You could use these to calculate concentrations as well, not just molarities. So in this example, let's say that I have a 100% stock solution and I want to prepare 2 dilutions from that stock. I could make, call it a 33% or 66% or 0.33 and 0.66 dilution factor. And I would use the same equation and calculate out the amount of methylene blue to add, and then use for any given volume that I want to look at. And then I would subtract that number from my desired volume to calculate how much of the water I would need to add to each one. Now sometimes I have a stock solution that is more concentrated than the solution I need. In that case, you want to dilute it to prepare your final working 1x solution. Many times all we know is how concentrated that solution is. So in this case, I have a 5x stock solution. It's 5 times more concentrated than I need. So I can again use the same equation to solve out for that volume and know how much of the volume of my stock solution I need to add to the working solution. In this case, it tells me that I would need 100 ml of the 5x stock solution. And that makes sense because it was 5 times concentrated. So I would take 100 ml of the stock and then add it to 400 ml of my dilutant to get my 1x working concentration of solution. Now that's one way to make dilutions. Another way is where we have a very concentrated stock or waste, and we need to prepare a super dilute working concentration that we can use to run our activity assay, or maybe we need to look at the number of bacteria or viral particles in the blood or in the urine or in the sweat, and we just need to dilute it by many fold, for like 10,000 fold. So if we would try to do that from that full strength stock, it's highly unlikely that we would get an accurate value because it would be a very small volume to pipette. Instead, what we do is we create what we call serial dilutions. So we take our full strength 100% concentrated blood or plasma or urine or waste or whatever it is that we've got, and we dilute it once with water or with saline to create our first dilution, let's say a 1 to 10 dilution. We then prepare another 1 to 10 dilution of that first diluted sample. By doing that, you suddenly now have a 100-fold diluted sample because you took a 1 to 10 diluted sample and you further diluted it 10-fold. We do that repeatedly until we get to our desired final dilution. In this case, it took us 4 steps to do that. Now this gives us better accuracy, and in addition, it also saves us waste. So we only prepare a small, tiny volume instead of making a very large volume of waste. Now once we make these type of dilutions, we can look at their accuracy and we can look at the amount that was prepared in each one by using a spectrophotometer. We can do it with colored solutions, but we could also do it with colorless cells, simply by looking at the cloudiness or haziness of the fluid as we increase the concentration of those otherwise invisible cells in a particular solution. The more cells are in suspension in a given solution, the more hazy or cloudy they will get. So when the light passes through that sample, a lot of it will get bounced off of these cells and get scattered away. So the more lights get absorbed during the course of the experiment, the more cells were there. And you will end up with a higher measurement with the spectrophotometer for the absorbance. So this just shows a spectrophotometer can be used as a colorimeter or as a spectrophotometer, where you just simply use light as your source. If you're doing it as a colorimeter, you're going to specifically look at light of particular wavelength. For example, if I wanted to look for blue particles, then I would just use the light associated with that. When we get the value from the spectrophotometer, it would be in the amount of light that was absorbed through our sample. The more cells are there, the more light will get absorbed, so you will get higher optical density and higher absorbance value. When we look at those numbers in a serial dilution, we find that those numbers are going to obey Beer's law as long as they are in the linear range of the curve. So the Beer's law says that the absorbance of light by any material is going to be proportional to the concentration of that material in the solution. We can look at it by looking at transmittance of light or through absorbance. In our case, we will focus on the absorbance. When we plot those numbers so that you have concentration of your solution or your cells on the x-axis and the absorbance that we get from the spectrophotometer on the y-axis, you would end up with a linear trend function. Now that linear trend function is going to be there for all subjects or all samples, except if you increase or decrease the concentration of your solutions or suspensions above or below the range of your instrument. So if you imagine this, there might be a situation where you only have, let's say, 1 molecule of yeast or 1 molecule of methylene blue per milliliter of water. That would be too small of a volume to be recognized by the spectrophotometer, to be detected by it, and you would never find it. So there will be unmeasurable difference in absorbance. That will happen for very, very small quantities or concentrations until you reach the linear range of the curve, where there is a direct relationship between the concentration and the absorbance that follows Beer's law. Similarly, on the upper end, you start to get samples so concentrated that almost no light is going through already. And so increasing the sample by, having twice as much is not going to have a linear or proportional effect on the absorbance anymore. And it will eventually plateau off into the maximum value that it can detect. Once we have the linear range of a particular curve, we can use it to create a trend function and a standard curve linear equation, a y equals mx plus b equation. The regression square of a linear trend function should be very close to 1. That will show you that that was a precise standard curve. It establishes the equation that you get, establishes a relationship between the concentration and the absorbance of the samples that you were looking at, and then can be used to find concentration of unknown samples. So once you have a standard curve depicting concentration of cells per mil with absorbance, let's say these are yeast cells, then I can be given an unknown sample of yeast suspension, and I can look at its absorbance and then use the equation to plot the absorbance in for y and solve for x and get the actual concentration of my unknown sample. So one of the things that we learned to do in this lab is pipetting. And we've talked about how it is very important to accurately pipette our samples. Just like we can use the spectrophotometer to look at the accuracy of our standard curve or accuracy of our dilutions that we prepared, we can also look at it to look at the accuracy of the pipetting technique that we are using. Typically, when we think of spectrophotometer, we think about how we use it to pass light through the fixed path length across the sample and look at the absorbance. You can also pass light up and down the spectrophotometer wells of our sample wells so that it is going through the liquid that is present within our each well. And this is looking at variable path length. The more liquid is present inside the sample cuvette or well, the longer the light will stay trapped inside these wells. And so you will get higher absorbance values when there is more liquid in the well and lower values when there is less liquid in the well. We use this to calculate our accuracy. If we repeatedly pipette the same exact volume in multiple wells, then all of them should give us the exact same path length. If they don't, it would indicate that we are not accurate in our pipetting technique when pipetting these volumes.